Great. So welcome everyone for the grand rounds. Today is going to be a day on new technologies, bioinformatics, machine learning, and a lot exciting stuff that we'll be hearing about in terms of its application for precision cardiovascular medicine. Before we get started, I'm going to share the CME and MOC credit slide set so that you can get situated with your credits. And for again, the YouTube website, you are very familiar where the most of the lectures are going to be there. For the CME credit for the session, 20407 is the number. Please text to 888-816-4893 as an SMS message. And for the MOC credit for physicians, it will be room code 2024. The link will be again displayed back, and you can go back. And a lot of you have requested that perhaps this link should be situated for more than 12 hours. We'll look into it on how we can extend our time so that you can get up in the morning and complete the link if need be. But the code is future 2024 and the link will be displayed. So step one, please complete your CME and the step two for getting the MOC credits. Thank you very much. With that, it's my great pleasure to bring in Dr. Zishan Ahmed, our grand round speaker today. Zishan is assistant professor on tenure track. He is now a faculty in the Department of Medicine, Cardiovascular Disease and Hypertension Division. And he also has co-appointment with the Rutgers Institute of Healthcare Policy and Aging Research. He was introduced to me, and in fact, he reached out to me last year, and I was very excited to note a tremendous potential that he has shown in his work here and also in the research work that you will hear from him. But I'm really excited about how he will be able to contribute to the future of the division, and particularly working with several of the faculty members. He may provide you a different direction in your research beyond the conventional statistical approaches in the electronic medical record-based data science that we are typically doing along with our fellowship program. Zishan has gone through a journey, and I'm going to mention that. He started from Punjab, Pakistan, 2005, where he did his software engineering in University of Central Punjab, and subsequently had his stay in Europe. He was in Sweden, Austria, and subsequently, and also at Germany. His PhD was in bioinformatics from University of Würzburg, Germany, on bioinformatics software for metabolic and healthcare data management. And subsequently, he came to the US and was at University of Massachusetts Chan Medical School for postdoc research. And then he moved upwards through Farmington, Connecticut in genomic medicine to have a seat as adjunct assistant professor and assistant director of bioinformatics precision medicine at University of Connecticut Health Center, Yukon, in Farmington, Connecticut. And he then subsequently moved here in 2020. Zishan has been contributing to several works, not only in educating in precision medicine and genomics and bioinformatics core, but also he has shown proficiency in extensive research, with over 70 plus peer-reviewed articles and extensive contributions as a co-investigator in several NIH research grants. I'm seeing several awards here, up to 14 that are listed. Many of them have NHLBI and NIH contributions. More importantly, he has been contributing to postgraduate and postdoctoral research, and several students are involved with him. So if you would like to get involved in student research and get a group of people who are enthusiastic with data and have a different perspective on how you can approach, that will be Zishan. Most recently, he has been also an entrepreneur and has submitted 2 patents, one of them being IntelliGenes, which I'm really excited to hear about, and maybe he will tell us about it in his work. With that note, Zishan, the floor is yours, and then we'll have a panel discussion where we will invite Dr. Yanmala and Chris Wang to join us for our dialogues.
Thank you so much for the kind introduction. It's a lot more than I deserve. So, with your permissions, I would share the slides.
Please go ahead. Yeah.
Just to confirm, Dr. Janamala is clearly visible.
Perfect. It's
Yes,
perfect.
everything good.
Okay. So, once again, thank you so much, Dr. Sengupta, for the very kind introduction. And my name is Zishan, and I'm very grateful for this opportunity to be here today and presenting in cardiology grand round. It's my honor, and the title of the talk is more close to the title of my most recently published article, which is Discovering Biomarkers and Predicting Cardiovascular Disease Using the Novel Nexus of Machine Learning Techniques. I would like to start my talk or this presentation by discussing one of the 3 causes of deaths in United States, and I would surprise you with information that medical error is one of those. Fair amount of literature has been published in last many years, which discusses medication error and delayed treatment in different ways. And one of the most recently trending subjects in the field of life sciences is precision medicine. Talking about precision medicine, this initiative has been first taken by our former President Barack Obama in 2015, and this is what the focus of my lab at Rutgers IFH and Robert Wood Johnson Med School is. Going a step further, talking about what is precision medicine. So it's mainly about, providing a personalized treatment to the patients, including their clinical profile, but also looking at their information, which includes information about their lifestyles, their genomic, or I would say multi-omic profiles, and information including about their phenotypes. So, more than me, you all know that when a patient, comes to a clinic, the basic steps involve diagnosis, different kind of lab tests, and if required, different medications are prescribed and procedures are performed. So one can ask a question that, with that, how can precision medicine contribute? How can it make things better in personalized treatment provision and predictive diagnostic? So to answer the question, I would lead with this question with another question, that one medication which might be effective for one disease or diagnosed, as a treatment for one patient might not be evenly effective for the other patients. And to investigate this cause further, we look into the genetic profile of the patients. We are interested in looking at the mutations deeply, and maybe there are some, important genes with significant expressions or both situations are happening. Talking about genes, this field is not new to us. It's been over 100 years since we are, living with this field and progressing a lot. In 1990, the first time gene word was, introduced, and then the story started. Then it progressed a lot with the development of new techniques for sequencing, DNA and RNA, and going forward, different kind of, human genome program is one of them. Different genetic programs were introduced. Different kind of huge amount, databases were introduced to archive huge amount of data. And most recently, we have started jumping into the era where we are developing, excellent bioinformatic techniques, classical statistics, and novel machine learning, applications and approaches towards the big data era. Talking about big data, in last couple of decades and more, huge amount of generic or big data or multi-omics data have been generated. So talking about data, this is a very complicated process, can be performed at one institution. However, most of the time it is, divided in multiple institutions located at one place at different places. So story starts with healthcare institution which is a primary, I would say home for the patients. and then different kind of clinical or research-based investigators start different studies to investigate different diseases and treatments and drugs, and they consult or have different collaborations with vet or bench to perform different kind of experiments. They connect with different cores to perform you know multi-omic data generation, most importantly sequencing data. Then different kind of IT-based environments are required, for example, high-performance computing and cloud computing environment to support the data management of high-volume data. And then, most importantly, we request the services of computational scientists and bioinformaticians who apply different kind of bioinformatics, classical statistics, cutting-edge machine learning techniques to process, analyze, and interpret the data and bring those results back to the healthcare institutions. I was telling you this all the story. Most of you already are aware of this. But the main reason to discuss this whole process is that every stage where the data was walking in, getting out, it is changing. So these are all the humans in different colors: blue, brown, black, gray, green, and purple. These are all different colors of same data. The data which was primarily was in the healthcare institution and walking through the core and coming back to through the computational science is different, and the time involved in different kind of data generation, analysis, and interpretation also consumed a lot. So this led us to the very complicated situation where we are not equipped with standardized data generation methodologies, standardized data sharing methodologies. For example, even today, many people or many labs share the data as Excel sheets through emails, right? So sometimes it is feasible, but in most of the cases, it is not. So with these all complications related to the data, the question is how we're going to do precision medicine, how we're going to answer those questions which are related to the identification of novel biomarkers and predicted diseases, how such heterogeneous data can be integrated, analyzed, and interpreted to support precision medicine, translational research in clinical settings, and variable research environments. So this is, one of the story above the sea. You can see that which is based on traditional healthcare data analytics, which provides a sufficient amount of information through the clinical data, which can support us, designing some studies based on diagnostic and treatment strategies. And however, this is not enough. And going forward, what we are looking forward is to generate, multi-omics data, of those patients whom we have clinical data. We want to fuse and integrate in a way that we can apply machine learning techniques, different algorithms, classifiers, which could be supervised, unsupervised, to discover novel biomarkers, predict the diseases or early diagnosis, and ultimately provide personalized treatment to the patients and address healthcare issues in an economic way. This is something I'm trying to study for last few years. I've been very fortunate to be able to publish different articles in the similar area, and a few articles have received some recognition and some citations as well. So if you are interested, please feel free to consult these articles. So this gives me an opportunity and a stage of my presentation to introduce you to my lab at Rutgers. What we are exactly doing towards the cause of precision medicine. We are a computational lab driven towards technology development. And the first kind of technology we develop is related to the high performance and secure computing networks. Because if we don't have the base, we don't have the technology to support the management, sharing, integration, modeling, and processing of the data, we won't be able to take a step further. Second is development of biomedical informatic applications. So biomedical informatic applications are required, most importantly, to extract, transfer, and load, the patient-specific, electronic healthcare records. We need to perform different kind of analysis using that data. In addition, we need systems which can help us in patients recruitment, for consenting and recruiting them for different, important studies. So we develop those technologies as well. Third, importantly, we develop bioinformatic applications, which includes tools, scripts, and pipelines and other technologies. It involves mainly because we are more driven towards generics. So we do implement different kind of tools and technologies and pipelines which can help us in analyzing and RNA-seq-driven gene expression data, whole genome or exome-driven variant data, and other relevant applications. Last but not the least, and most importantly as well, we are focused on developing machine learning, approaches, to to improve the methods so we can try to find information or the patterns revealing predictive and modifiable risk factors to support early diagnosis of tailored disorders and sequelae. So we use these all four different kind of technologies ultimately to support multi-disease specific research. So talking about disease specific research, I would like love to take you the disease which is close to my heart, and that is cardiovascular disease. Talking about cardiovascular disease, I feel like a student in this seminar. You all know much better than me, and but I still would just mention that briefly. That we are focused on heart failure and atrial fibrillation, which are among the most common manifestation of CVD and contribute about 45% of all CVD deaths. So, going deeper into the investigation of cardiovascular diseases, we have set some challenges and some goals to meet. So, the first goal is to identify genes which are known to be associated with heart failure, atrial fibrillation, and other cardiovascular disease based on the state of the art literature. Our colleagues in the world, in the US, who are doing this research for the last many years, we wanted to learn from their work before we jump to any conclusions from our own analysis. Next, we wanted to investigate genes associated with these disorders based on the orthodox and standardized considered bioinformatic technologies, for example, gene expression, enrichment, pathway, functional mutation, and other analysis, most importantly, GWAS and other tools as well. Then going forward, we want to make some improvements as well in the analytic side with the implementation of machine learning approaches to predict the cardiovascular diseases. However, using known biomarkers based on the analysis we have performed and based on the information we have received from the state of the art. Fourth challenge, we want to discover novel biomarkers and predict disease with high accuracy using complete multigenomic profiles integrated with the phenotypic profile. This is something new in the world. Not many people have excelled already in that, and people are trying very well as well at the same time. Last but not the least, validating results produced using machine learning techniques for the translational research. I always, believe that why should a clinician buy my results? Like why they should trust on the results we have produced using machine learning. This is a very, very important step, and we we do that as well. And how we do that, you will see in the upcoming slides. So I would introduce you to, or I will walk you through to one of the examples we have done at my lab, and that's a study we have done. A small step we have taken to learn actually cardiovascular diseases. In this small study, we started and took the first step by learning from state of the art. As I said, we designed a basic infrastructure. We developed some mechanism to extract healthcare records. We did some sequencing. We performed different kind of gene expression and variant analysis, and we also performed predictive analysis and discovered different novel biomarkers. So going to the next slide and starting, and explaining this workflow, I would start with the learning from state of the art. So I, assembled, a group of students who worked with me for this project, and we started learning, in two different ways. Number one, disease-specific goal, which is definitely based on heart failure and atrial fibrillation. Second goal was to look at the existing bioinformatic approaches, which have been specially published within last 5 to 10 years, and we were interested in identifying how many genes have been reported, and we found over 190 genes associated with atrial fibrillation and cardiovascular disease. Then we compiled an interesting study on that, and we were very fortunate to be able to publish this article as a review study. Last year, it came online because peer review processes take some time, and there are some conclusions which you can see, in this figure that how many articles were reporting how many genes and what are those kind. So going to the next step, one of the important, thing for us was to design a supporting infrastructure, because we were very fortunate to have access to the high performance computing environment at Rutgers by OARC. Then we were very fortunate to have access to the secure environment provided by Institute for Health. And then we also developed some new servers. So what we wanted to do that, how can we efficiently link the data together? So we utilized a data modeling approach within high performance computing, where we processed all the raw high volume sequence data of the patients. Then we, connected those pipelines to the databases or data management systems deployed within the Institute for Health web servers and database management servers, and the data was automatically transferred. The final outcomes were automatically transferred and linked with those databases where there were already patient diagnostic information and treatment-based information in a de-identified manner available. And then we also deployed a connected web sharing server. Like, so we have developed some applications and bioinformatic tools and websites where we are, sharing some data as well, so that those applications support us or supported us or continuously supporting. And I would proudly say that some of our applications have even more than 1,000 users in US and worldwide. So going forward, I would say that, to support that study, the first step was to collect information from electronic health records of the patients we would like to consent with. So we designed a new tool which we call MapClick, because as Dr. Singupta explained, I was in Connecticut in Yukon Health. There I started learning how healthcare systems work. How can we develop some applications which can be plugged into the back end of the healthcare system, so we can pull some important information based on the patient's diagnosis, their personal information, their visits to the practices, and their treatments which have been provided throughout the time they are coming and since they are coming. So we developed the tool which we call as MapClick. This is an open source application. We presented in EMEA and published with the JAMIA, the journal by EMEA as well. So it was very good for us to get that acknowledgement. And then we took a step forward to enhance our system to support patient recruitment. On the left side of this slide, you can see there's a small workflow which explains how does the system work. Like, the patient comes to the home healthcare institution, they're connected to the Epic system, which is containing their electronic healthcare record system and information. Then all those patients who consent for our study, we have access through this tool MapClick to their electronic healthcare records. Then we remove the identifiers from the data and make this data completely de-identifiable. So it's not. And then we use that data to build the disease-specific cohorts, and then we go next level and collect the samples from those patients for the different kind of experimentations to support translational research. For the study, as an example, which I'm presenting in this seminar today, you can see in the bottom there is a small bar chart which is representing a population which is based on 61 patients with cardiovascular diseases and 10 control patients. So the population which is between 648 to 658, these are the controls, and the population which is between 1058 to 1118, these patients are diagnosed with cardiovascular diseases. And there are different kinds of races involved: white, black, male, and the different genders, male and female, definitely. So we took these, we consented these patients, we took their blood samples, and then we moved forward to sequence it. So we performed RNA sequencing, to study the transcriptome, and we performed whole genome-based sequencing to study their genome. So on the right side, for those who are not familiar with the genomic world, this kind of reports we generate to assess the quality of the data. So how the data looks like, how much percentage of genome is matched to the known human genome, and same goes for the transcriptome. So the yellow color is aligned to the genome, but you can see the percentage is close to 100% in terms of the data quality, and purple is the transcriptome. So we, once, and the good thing is that when we were doing this sequencing, we sequenced same patients for both kinds of data generation. So I would like to start first with the gene expression, like the RNA-seq driven gene expression data. And for that, one of the challenges I was facing, that when I joined Rutgers 3 years ago, I was able to do that. But COVID hit that time. I was not having a very good support in terms of having good students to support my research. So I was confused a lot. I thought, what should I do in that situation? Should I do all the analysis myself? Or should I train the students to do the analysis? And if I have to train the students, then from where to start? So then I thought, okay, let's go and develop a tool, which can help students in doing gene expression analysis. Then by my lab, we produced, a tool which is supporting RNA-seq. And just to explain, RNA-seq has become the most used method for gene expression analysis in the world. And that's why we were very much interested in that. And we thought if we can develop a tool which is more acceptable to the community, a lot of other people, same like us, can get benefit from here, from this tool. So we developed this tool we call GWiz, and it is a fair application that I call fair. It means it's a findable, accessible, interactive, and reusable application. We developed at Rutgers, and it supports transcriptomic profiling and expression analysis to identify, admire, and compare genes and transcript in multiple conditions and in different tissue and individuals. So going forward, I would briefly explain you how this tool works for us. Maybe if you are interested in collaboration or if you would like to use it, it might make sense for you. So it connects to the database, which includes all the gene expression information. I hope you remember the slide I shared, 3D, like three kind of servers. This is connected to the database server. Then the second step is to select the data, select the expression data. For that, the question could be the how expression data have been generated, because the raw data we received was in FASTQ file. So for that, we developed a new pipeline at Rutgers, and we called it this GWiz RNA-seq data quality checking and processing pipeline. This pipeline is not a novel pipeline. This is orthodox pipeline. A lot of people in the world are trying to get benefit from similar pipeline. However, we have developed this customized pipeline ourselves. We have not downloaded any source code from anywhere, but programmed it ourselves. And according to the customized, servers at Rutgers. And this pipe, the beauty of this pipeline, or something which makes it different in comparison to other pipelines, is once all the data processing is performed, all the information about captured isoforms and genes is automatically parsed and loaded into the database and back and connected back to the patient's healthcare record. So, for example, if you look at a clinical record, you can have the patient's diagnosis, but you won't have uh, information about the genes related to that particular disease, and their significant expression could be higher or lower. So with this kind of pipeline, you can connect there as well. So going next is the gene annotation step. So now you have information about the genes, of the patient, but you don't know which gene is connected or associated with which disorder. So that we started another project in my lab that, I have some students which started working with me. So we went to, I give you a situation. I say, okay, there's a gene BRCA2, one of the world's famous gene. If you type that in the Google, you will find, maybe 100 pages of the Google, which can tell, okay, this gene is linked to breast cancer. And there are so many other websites which are providing you this gene-based information. So if you, go deeper and deeper, you will see, and you try different genes, you will be slowly starting confusing that which source is more authentic, which database is more authentic to consult. However, there are some big names like CleanVar and some others as well to rely on. So this question we asked as well a few years ago. So we started looking at all the authentic listed databases, and we ended up having over close to like 80 databases and tools available which claim to provide gene disease information. So we compiled all those database based information. We compiled all the hard work we did in a manuscript which we have published in Briefings in Bioinformatics, as well as we compiled a new database where we integrated all the freely available databases, the distinct information. All information was related to gene, disease, SNP, and disease. So having a mutation in a gene, which disease have been reported, and having a significant expression in the gene, which disease is reported or which diseases are reported. So then we thought, okay, this is good for us for personal analytics in the lab. However, we could share this with the community as well. So we designed a small smartphone application, which is freely available on Apple stores. You can download it for education and research purposes, and there's no cost on that. We also written an article on that, and we published in a handsome journal and shared this database and this app. And this is one of those applications where we have more than 1,000 users and students involved. So going forward, we also took another step recently. Last year, we published another article there is an institution, which approves, genes for the medical use, which is called ACMG approved list of genes. So according to the last year, when we closed our study to a step, there were 73 ACMG approved genes. So the challenge over there was that we were aware, okay, these genes are linked to the diseases. However, to do the translational research, we need to link the diseases with their relevant ICD codes as well. So we try to find if there's a database exists which can help us precisely linking genes to the disease and that disease to the relevant ICD code. And you all know much better than me what ICD code is and what are their types, 10, 11, 9 different versions we are having. So we started manually curating the database and we published one of the good journals by Oxford where, this our database where we have linked gene diseases and their relevant ICD codes. So we used we integrated these all databases in our GWiz. It was just to give you a background. This GWiz tool for gene expression analysis is also published and open source and freely available. We were very fortunate to be invited by one of many different conferences, which includes the top conferences of the world, especially in the field of bioinformatics, which is ISMB from ISCP. I was the speaker over there. And then I was also invited in EMEA conference, Informatics Summit, for presenting this tool as well. And we also conducted a few full day and half day workshops at different international places to educate more researchers about how to use our tool. And there's a YouTube video as well if you want to familiarize more closely to yourself. So coming back to the study design, I just given you the background. This tool was developed. So using this tool, these are some examples of the outcome produced by the tool with some click operations. So you can see in this figure there are three figures. These are called heat maps. So the first heat map is about the genes associated with heart failure. Second is atrial fibrillation, and other if there are any other cardiovascular diseases which having significant expression. So all what you need to do using this tool, you need to select the patient population and you need to select the disease, and then you can even select for the genes as well from the significant, non-significant, and all genes. And then you can perform gene expression analysis and you can identify all those genes which are having significant behavior or expression or patterns in the disease relationship. So in this, I just to a little bit elaborate or you on that, you can see the top figure, which is blue and red. On the right side, you can see the pattern is completely different of the gene expressions for the patients with no CVD disease reported. However, the patterns for the CVD patients is much red and less red, sorry, and different. So same goes next. Like we this tool can help us in categorizing or classifying the genetic information based on the gender. We can look for the male and the female based significance in genes, and we can produce different kinds of heat maps, including information about the genes, for example, on the left side, on the right side, for example, information about the associated diseases, and on the bottom, the samples we have used. It can help us in looking at the protein coding genes as well as non-coding genes as well. Then it can help us in classifying further based on the race of the patient as well. We can classify here for this example, we have white and black because those were the heavy population. If you remember the slide I showed a few slides before, where we were using MyClick to consent the patients, and I have showed you a bar chart over there where we have race and gender-based classification of the patients consented for the study. There we have dominating race based on white and black, and all others were also there, but we have categorized them as one. So what we did, we wanted to, kind of perform differential gene expression analysis as well to validate our results. We wanted, in another way, I would say, we wanted to also perform orthodox bioinformatic analysis to see if our results produced using GWiz are correct and can be validated through orthodox approaches. To validate that, we perform traditional differential gene expression analysis, which clustered us in different important genes. And then we also perform different kind of gene enrichment and pathway analysis, which has given us a good amount of validation of the results we have produced using GWiz. So conclusively, we were able to kind of publish two interesting studies. One study was completely based on the patients, for reported for heart failure. Other disease was more close to atrial fibrillation. And the findings I could summarize for you that like we were able to identify over 4,000 differentially expressed genes. 15 of the genes were significantly reported, and we have found some highly expressed genes in HF, some highly expressed genes in AF. Right, we found some gender and ethnic group specific analysis, based genes, and also subsequent analysis were performed based on gender or analysis identified altered expression pathways of genes with gender differences in middle age to trial CVT patients. So these two studies are also published, just in case if you are interested in more having information about that. So going forward, we wanted to see, okay, the first question was answered. We wanted to investigate the expressions of the genes related to the heart failure, atrial fibrillation, and other CVDs. But we wanted to investigate their variants as well. So if you remember, I told you in the beginning that we sequenced to up two kinds. We've sequenced their RNA, we've sequenced their DNA, like whole genome sequence data. For whole genome sequence data processing and analysis, RNA sequence data based pipeline cannot function. So we wanted to produce a new pipeline which could help us in discovering the variant space information. So this was another pipeline we produced at Rutgers. Again, this is an orthodox pipeline. It's not something we have invented, but we have definitely re-implemented, customized to the settings we have at Rutgers. Just in case if somebody would like to use it, it's already available in the data cluster. They can just call it and use it. So going forward, we applied this pipeline to the same samples, same patients, whole genome sequence samples. We try to identify and look at the percentage of the mutations, in genes for related to the heart failure and the other CVDs. We perform different, analysis, which includes mutation analysis as well for the gene associated with heart failure and atrial fibrillation. We performed variant and prevalence analysis where we try to see the distribution of the variants or the variants among the genes of our interest. We also performed some basic classical statistics as well, where we use JST method for the investigation. And then we also went further and performed a functional and non-functional mutation analysis of those genes which we have discovered with significant expression during our gene expression analysis using GWAS and other orthodox bioinformatic techniques. So we also found some interesting results, which were based on varied mutations among the genes. We detected over a million SNPs, and and insertions and deletion events. We identified some interesting genes as well. And the most common mutation type in CVD-based, diseases were, the genes with intronic, were intronic, and mutation in these genes have been made to aberrant expression in CVD. So we were very, also fortunate to compile these, this, all the findings in one of the primary studies published earlier last year. And then we consented 35 more patients and tried to reproduce the similar results which we have produced in the first study. So then we published another study which is in scientific reports this year, last year as well. So jumping to the almost last part of my talk, and that was about how we did implement machine learning, artificial intelligence and machine learning. So when we try to implement machine learning, the first step is that, how are you going to do that? The data which you have, received from your electronic healthcare records, or the data you have received from different, cores, cannot be directly used for any kind of machine learning analysis. So we need to, perform some data customization or restructuring. Then, our goal was to predict the disease with high accuracy, and at the same time, we wanted to identify novel biomarkers. So, that was our goal going forward. What we did, we designed a new study, which you can see on the screen. So the first step was to restructure the data in a way that we can use that data for deep phenotyping and predictive analysis. The second step was to identify novel biomarkers, which are currently not part of the existing literature. So what we did, taking a step, as I said, I always believe on not reinventing the same wheel, right? As a group of, I have a group of students who who started learning about existing machine learning and artificial intelligence-based techniques, which have been heavily applied for gene expression and variant data investigation to support personalized treatment. So we discovered variable, studies reporting the utilization of variable machine learning algorithms in this interesting picture or figure. You can see at the bottom, we have different machine learning algorithms. And at the top, we have different kinds of data sets. And in the middle of part of that, different kind of algorithms are, interacting with different kind of diseases. It could be cancer, it could be CVD, another disease, Alzheimer, etc. And we are trying to see how effective they are. The most, used algorithms found to be random forest and support vector machine. So this was our starting point to implement machine learning techniques. So we first designed a new pipeline, which we called as high gear, where we started with, random forest and then support vector machine. We implemented this pipeline and applied to the data we have already analyzed using orthodox bioinformatic techniques. The goal was to, as I said, to use the known biomarkers and try to predict the disease at high accuracy. So we were able to publish our results in another article last year, which is in genomics, and the article was summarizing the conclusions of the article. I would say that during the analysis or machine learning analysis, we observed that age and gender appear to have a high correlation in heart failure and other diseases, while age and race were having high correlation in atrial fibrillation. And we went further and further and reported some different results in this article. So just mindful of the time, I would say that we also published this pipeline as an open source application, so other people can take benefit from our application. So the last step for this talk is to identify new biomarkers. So we were interested in exploring the whole transcriptome and the whole genome of the patient. So if you look at this small abstract figure in this slide, you can see the first layer is the complete genome, the second is the target genome, and the third is the remaining genome. So it's same. If this situation can also be applied for the transcriptome. So when I say target, then disease-specific genes we study. So in that situation, most of the parts of the genome or human genome, I would say, or transcriptome, are left unattended. We are not aware what is going on over there. So we wanted to study or implement an approach which can help us in investigating the complete transcriptome of the patient, completely unbiased, starting with the supervised and ending with the unsupervised approach. So we designed this new approach, which Dr. Singupta briefly mentioned as well as like intelligence. First, we tested different algorithms, different combinations, and one of the combination which stood top was about this. So where we have, first of all, created a new data format, which is called CIGT format. And interestingly, since we had this publication out, we have already fair amount of citations to our data format as well. And that was pretty surprising that people are more interested than the approach in data format. We use the data format, we went the data, we pass the data to the different four different tests to identify novel biomarkers, and then combined list of markers together with the test and the training models. We used or utilized four different kinds of machine learning algorithms, which are like including random forest, support vector machine, XGBoost, and k-meter stable. The outcomes of all those four algorithms were merged, and we use different classifiers and score ranking techniques to identify the most susceptible genes from the overall machine learning model. So, these are some results we received. These are the four algorithms, I would say, biomarker discovery algorithm-based results we reported in the article, which have been published already. Then we performed, the goal was we wanted to see, okay, there are hundreds of biomarkers we were able to identify, which were common among these. But what are the most common and significant biomarkers among all of the algorithms and classifiers we were implementing? So we were able to identify four different important markers, which are in the bottom of this slide. You can say MTR, NR2, L1, LILRA2, and there was one novel protein which was not even in the literature, and then HLA-B, which is the most commonly reported gene. Then going forward, we were interested that these genes are related to which diseases. So we were able to identify in this network diagram. You can see all the squares which are in orange color or red color are CVD, are representing cardiovascular disease. All the boxes which are in blue color, those are representing non-CVD. It could be diabetes, it could be Alzheimer, it could be any other disease. And all the circles in green, those are the genes. 18 genes we found as novel biomarkers, and 4 genes we found as common biomarkers reported from all approaches. So going forward, we went back to the electronic healthcare records of those patients because we wanted to see, we are discovering, like for example, in one of the cases, we found a patient with breast cancer. So we were not aware that patient has breast cancer in the past or not. But we found gene and mutations which were telling us, guys, this patient has breast cancer. So then we went to the electronic health records and we were able to identify and validate our results. The patient has been reported for the breast cancer. Same goes for different other diseases as well, which we found from the genetic perspective, but we were not having already knowledge about those diseases relevant to those patients. And these are this figure is part of the paper as well. So I would conclude my presentation by saying that this whole study is getting some reputation, and I'm so fortunate for that. It's been published in Scientific Reports this year, most recently. You can have access to it, and all the methodology which we exercised on these patients we called as IntelliGenes. The first version of IntelliGenes pipeline have been published in one of the top journals from bioinformatics by Oxford University Press, and that's also open source, freely available. Second variant we are developing and hope to make it available soon in a peer-reviewed publication as well. And there's a small YouTube video available if somebody is interested in learning how intelligence can be used in a short span of time. So before I end my presentation, all the work I have presented is because of the hard work of my students in my lab and collaborators. So I would like to thank my students for their active participation and when they were alumni. And thank you so much, Dr. Sugupta, again for the presentation and your support for the last few years, and all my other collaborators. I'm not very lucky to have big grants, but I have some support received, which is so far helping, and looking forward to more funding opportunities. And I would love to have any questions if you have. Thank you so much again.
Thank you very much, Jishan. So we're going to have a panel discussion now. We have Chris Wong, who's joined us, and he's one of our fellows, and Dr. Yan Mala, of course, we all know. And before we turn it over to Chris and Navina to have a discussion with you, thank you very much for bringing this up for us. I'm going to mention one thing which is more central to clinical division. Clinical cardiologists find the language of machine learning and all the words that you're using pretty foreign, because in our routine clinical day-to-day work with our patients, we hardly use this words. And if I understand correctly, and I'm taking the, not that I don't understand AI and ML, but I'm going to imagine myself as a clinician. So when do you think can we start applying? Let's assume your work with IntelliGenes, which predicts the presence of cardiovascular diseases. When do you think we can start applying, or in what form and shape we can start applying? Could, for example, children of patients who have coronary artery disease or relatives use this to understand what is their risk for future developing, or how do you propose to apply this in clinical medicine? Is it ready for application right now?
Thank you so much for this great question, Dr. Singupta. And that's actually the reason we are doing this research, that ultimately we can bring these kind of technology to the clinical setting so it can be realistically applied to support translational research. Definitely, this tool in current form we can apply, but with the help of bioinformaticians. Recently, we have started developing a new tool which is including a graphical user interface. So we are testing these days. It's getting closer to the final stages. That tool can be interactively used. When I say interactively used, to be precisely explaining it, you can use, some mouse click buttons to do the process and analyze the data of the patients and identify the biomarkers and predict the disease. So based on the factors we have reported or results we have achieved, definitely with some more support from the clinical division, like for example, Dr. Sangupta, you said, Zishan, We have data available and related to the patients, and can you bring this tool in and try, this to the data available? That would be great, and that's what we are looking forward to to experiment with the data available through NIH channels as well. So there is a new archive available, biodata catalyst from NHLBI institution. So we are looking forward to download some data sets from there as well. So the goal is to try this to the maximum data available before we can declare it that can be used for translational research in clinical settings. Because a big risk if we produce some results which are not accurate for any reason, or maybe we have not discovered that problem right now, then it can be life-threatening. So we cannot rely on that. Definitely, we are in experiment stage right
Who wants to go first? Maybe I'll start with Navina. Navina.
Thank you, Jishan, for the presentation. It's really encapsulates what you have done at Rutgers for the past 2-3 years, and it's captivating. And my question is with the integration of your findings and biomarker discovery for CVD and its combination with clinical data. AI and explainability is becoming more and more relevant when it comes to machine learning to adapt and implement in clinical space. So how do you explain the relevance of your findings with the clinical data? And have you actually did some analysis or research with the classical clinical risk factors and the genes relevance? Number one. And what molecular pathways? I know that you have shown me that when you went back and digged into the EPIC, you did find association with the genes. But usually just 1 or 2 genes is not enough, right? A molecular biological pathway is important for explainability and understanding of the disease process. So
Yeah, so the first question, like definitely that is the goal, to start and end the journey into the clinical data. So that's why, we were very much focused on the use of clinical codes, because each disease has several phenotypes, or branches. I can say diabetes, but for Dr. Singupta and Dr. Wong, they might ask which diabetes you are exactly talking about, right? And if there is some disease related to CVD development, then it's further complicated, right? That's why connecting right information at the right stage is very important. That could only be achieved, at least today, with the use of ICD codes. So that's why we use different factors. We use personalized factors based on their gender, based on their race. We use clinical factors based on different phenotypes of the diagnosis. If you will see that paper, you will see we have reported not 1 or 2 genes with that. We have reported more than 700 genes. We found which could be used for the multiplexed pathways. But we have reported 18 more significant. So one thing what we did, which in something I'm going to put on the table, which have not been published yet. So we have done this on transcriptomic data, but then we have again investigated this whole process using genomic data as well. That study is not published, and there are very few studies in the world. In CBD, there is no study in the world right now exists where they have investigated the whole genome, right, integrating the transcriptome and integrating the clinical profiles of the CVD patients. There we have found, the funny thing is that maybe next time I can discuss that, we have found not only the regulations in expressions based on, the transcriptomic data, but also the mutations. And then we went further. That is a very complicated system we have developed already, but we are practicing, testing, and we cannot declare. I myself not fan of being you know producing some news which is not authentic. I want to test my approaches on at least 10 to 20 different data sets with variable variations, and see if our system works in different conditions, different data types, different clinical features involved. Yeah.
So, if I add one more question, the cognitive burden of clinicians is a major discussion we think about when it comes to disease diagnosis and subphenotypic risk stratification. So, genomics itself is so big, 40,000 genes we are talking about, then combining all the multimodal layers of systems biology. And thinking of the other hat, association versus causation, right? That's very, very important. Are these biomarkers predictive or diagnostic? Could these be used for monitoring disease progression? What are your reflections on it?
Definitely, definitely, these could be used. But as I said, just like for me, it's like a network. You pick one node and then you rotate the whole network. Then you go and pick another one and then you rotate the other. So every node becomes a perspective for you, the way you look at the rest of the network, right? So, it highlights the spots. So you are absolutely right. I agree with you. At one side, it's very complex. Second, it's dense, populated, high volume, and one more thing you might ask, we have not discussed ethics. These days, I'm also working. What are the ethical problems? If we use genetics information. So that is again another top story running by NIH for last few days and weeks. How can we help in implementing machine learning techniques on genomics data without compromising the ethics? So that is, I agree with you, but we are on it, and I think there are more things to cover in the future, a lot more.
Thank you, Jishan. Yeah, Chris.
Yes, thank you very much for such a wonderful talk, and congratulations on all of the success that you've seen. This is really astonishing and amazing research. I think you alluded to it at the beginning of your talk, but I'd like to get your opinion on, I guess, to piggyback off what you just said, the ethics of the situation and how we maximize as a scientific community data sharing while also respecting people's privacy. So do you see data sharing happening at the level of sharing the patient chart data and genomics? Or do you see it as sharing some sort of abstraction of that? And if it's the latter, could you talk a little bit about what abstraction from it that protects patient confidentiality you might see as the most feasible? Or if it's the former, could you describe what sort of political buy-in we would need to see that out in the coming years?
Thank you so much for a very good question, very important one. And so I would say, and you mentioned those achievements. Actually, those are just small milestones we are trying to achieve. Nothing achieved as yet. It's just, we're trying to progress. Anyway, so coming to the point, which is very important. So there are two ways of, to the best of my knowledge and abilities, I would say that there are two ways of, protecting this, information and helping this issue. When we talk about clinical data, de-identification and data privacy protection is addressable. We can knock out those elements which affiliates, the clinical records with the patient's personal information. However, in genetics, it's not easily possible. One of the issues, like for example, I was even today talking to some of my students regarding the ethics. He was talking about data de-identification. I said, what if the genetic data of the whole city is available? By analyzing the genomic information of the patients, you can easily identify families, right? And there are more and more points which I can go in depth to give as an example, to avoid the breach. So this is a big concern. A lot of data, or I would say tons of data or tons of generic information have been already shared through variable archives. So this is the biggest concern. Earlier, just if you look at my 5 challenges, the first 2 challenges of my study was based on orthodox approaches. Using orthodox approaches, it was not easily, achievable, or it's not easily to compromise the privacy issues or ethic issues. But with the use of AI, you are letting the machine do the job, right? It's more dangerous. That's why people are very much concerned about that use of AI on genetics, that it will not only solve many problems, but it could create new problems as well. So, so that's why it's very, very important to address data ethics in genetics.
Zishan, I wanted to just pivot towards an important topic. So what are your resources, or in terms of, I think you have access to some databases or work or ongoing projects. How could our fellows in cardiology get exposure to be able to work with you along with Naveen and others to create more research in areas and more abstracts? Particularly, how could the division benefit from your presence and create more research in this area? And I think in the future, I would like some of my own colleagues and fellows to work with you to put those kind of articles that you're writing. How could we do that?
Yeah, definitely. It'll be actually my pleasure and honor if that is possible.
So
Because,
get ready, because like they are very dangerous. They will come in like all swarms, and they will
no,
go and they will go around you, and then I'm just joking. Yeah, because I think they're very hungry to do more research. So
so the thing is that, this is now a global world. Everybody is, the more connections, the more collaborations, the better success will be. So I am a student of science. I consider myself student of science, and I personally or honestly know nothing about cardiovascular diseases. I'm just a beginner, I would say. So, integration with the experts will definitely not only help growing my skills and exposure to the field, but it will also help me in understanding the complexities and the problems clinicians have, and I'm not aware of. This is one side, definitely. Second thing is definitely the two sides of the coin, I would say, when we talk about genetics. One is when we directly work, just like Dr. Sengupta, if I work closely under your supervision, you can help me in connecting me, if not directly to the patients, but indirectly to the records or data or genetic data, right? So we can use, if there is some data available, or we can produce the sequence data of the patients. We already have the samples collected, or if this has not been even done, then we can collect samples of the most critical patients or important subjects, sequence them, and then drive new studies, right? And we can start with pilot studies. We can invite, or we can write different interesting grants, to get more funding to do the same operation or same analysis for the similar other patients at the large scale. This is one way. Second way is validation of the results, because one of the complications we are facing with AI is reproducibility, how much the results are reliable. For that, the diverse data, variable data, and in some cases high and in some cases low data is important. For that, I can definitely help the division in a way that I am trying to learn and explore different NIH hosted systems. One of the newest system is the biodata catalyst. Actually, this system was introduced to me by NHLBI themselves. So I was connected for a grant with the program officer. She was so kind to introduce me. She said, Zishan, you are going to use TopMed. Why don't you go to the TopMed through the channel of Catalyst? Then she introduced me this, and then since then we started looking in more in depth that NHLBI offered this system, what this system exactly does, and the data they have stored over there, according to my today's information, is in petabytes. So it's a huge amount of data. But the problem over there is that there is some structured element. There are some structured elements. There are some raw data files. Naveen, I can understand junk, data like FASTQ dump, BAM, SAM, BAD, every kind of data is dumped. That kind of situation is very complicated because we cannot easily understand what's going on there, what kind of data they have generated, what kind of pre-processing they have done, what kind of pipelines they have applied, what final results are there, is those results are targeted or the complete unbiased or biased. So I can definitely help growing the research side by giving an introduction to these important archives where clinicians, can get some exposure that this is something which is coming from the US institutions in terms of genetic information or genomic data sets or multi-omics, I would say, data set. These are the studies are published. So we can even be part of some big, networks from genomics side, I would say. And these are the two different streams I can definitely explore. One related to the direct patients associated with cardiologists or experts in division. Second, giving exposure to the publicly available information from the multi-omics side.
very good. Any other questions from either of you?
Out of
Hi.
Singapore. So Jishan, if you can actually put together databases that you have access to and description, a small description about it, I'll be
Yeah.
happy to circulate in the division.
Yes, I can tell you that a lot of people here, I'm going to encourage everyone to come together. Still, I'm going to feel like the feeling is like, Einstein's theory of relativity and the electromagnetism, two worlds completely different. Coming together is a lot of difficulties and a lot of theories do not work together. So we have the physical world of a cardiologist, and then you have the bioinformatics core of and heart of database or genomics, and these two worlds we need to start bringing together, and that's going to be my appeal to the people here. And hopefully my wish list will be that next ACC we should have at least half a dozen papers or abstracts which are exploring some phenotypes of known diseases in cardiology at our institute or elsewhere with some of these genomic databases to develop either association mining or pathways. So let me see who can pull up to this challenge.
I would add one point to that, Dr. Singapore, and that is because you are going very high in technology development, especially when it comes to biomedical imaging, right? So if our institution or division is noticing some important patterns, we can definitely investigate if we have,
Yes.
tissue specific samples, that if there are some important expressions and mutations relevant.
Yeah, our phenotypic expression of the diseases will be very bright side. And I think bringing that with the genomic will be a very good aspect of the division to explore more. With that note, thank you very much, all of you, and we'll look forward to continuing the collaboration. All right.
I'm
Bye-bye.
grateful for the opportunity.
Yeah,
Thank you so much.
Bye.
Thank you, everyone.